Hello, my dear students. Today we learn one of the most important topic, molecules or mycoplasmas. Generally, we know the living organisms are divided into two types. First one is prokaryotic, second one is eukaryotic. The eukaryotic organisms, a conspicuous nucleus is present in eukaryotic organisms. But however, in prokaryotic organisms, conspiration nucleus is absent in prokaryotic organisms. So these are the main difference between prokaryotic and eukaryotic organisms. So in the prokaryotic organisms, in past days, they are differentiated into two types. First one is eubacteria. First one is eubacteria, and second one is archaebacteria. So in past classification, the prokaryotic organisms are divided into two types. First one is eukaryotic organisms, sorry, eubacteria and archaebacteria. But in the latest, Carl Woese proposed three domain system, three domain system. In this three domain system, first one is archaea, first one is archaea, and second one is bacteria, bacteria, and the third one is eukaryote. Third one is eukaryote. So these are proposed by Carl Woese. Three domain system. It is popular archaea and bacteria and eukaryote. So under the bacteria, we discussed one of the important chapter, that is molecules are mycoplasmas. What is the major definition of the molecules or mycoplasma? It is very simple and easy. Which bacteria we called molecules or which bacteria we called mycoplasmas? Generally, bacteria covered with a special layer that is called cell wall. So the bacteria are covered with a special layer that is called cell wall. Cell wall. Which bacteria do not contain cell wall? Most important. Which bacteria do not contain cell wall and must belongs to gram-positive bacteria? Now they are called molecules or mycoplasmas. So there are two important characters of the molecules or mycoplasma. First of all, cell wall is completely absent in the organisms, and mostly they have gram-positive bacteria. Absence of cell wall, and mostly they have gram-positive bacteria. Now they are called molecules or mycoplasmas. So these are the major definition of the molecules and mycoplasma. So mycoplasmas are molecules into which class of bacteria. So class of bacteria, firmicutes are bacilliota. So firmicute or bacilliota. Another one is gratiludes. So gratiludes are belongs to gram-negative bacteria. Firmicutes or bacilliota belongs to gram-positive bacteria. So firmicutes means firmin strong, cute means skin. So firmicutes are gram-positive bacteria. Mollicutes are one type of firmicutes are bacillata. Why? Because mollicutes are gram-positive bacteria Mollicutes are gram-positive bacteria, lack of cell wall, most important, lack of cell lack of cell wall. So, mollicutes are one type of firmicutes are basileota. So, we discussed 1.1 another. The general characters of mollicutes are mycoplasmas. Mycoplasmas. So, the mollicutes represent a group of eubacteria. Mollicutes are belongs to eubacteria. The common name of the mollicutes are known as mycoplasmas. Mycoplasmas, not mycoplasma. Mycoplasmas. Mycoplasmas. So a lot of controversy arised mycoplasma and mycoplasmas. Mycoplasmas is a general term. All gram-positive bacteria without cell wall, all are popularly known as mycoplasmas. Then mycoplasma is one of the subcategory in mycoplasmas. A lot of controversy arised. So that way the mycoplasmas are changed the name into mollicutes. So mycoplasma or mollicutes are the smallest among the aerobic prokaryotics. So mycoplasma are the smallest among the known aerobic prokaryotic. The lack of cell wall, the mollicutes, gram-positive bacteria and lack of cell wall, so they change their shape into different types. So mollicutes are mycoplasma pleomorphic. Why? Because some of the oval shaped, spiral shaped, rod shaped, comma shaped, so they change their shape every time. So mollicutes are pleomorphic in shape. These mollicutes or mycoplasmas are first discovered by Patcher, but he was unable to isolate them into pure culture. At that time, he called pleuropneumonia-like organism, PPLO, pleuropneumonia-like organism. Later, Nocard and Raux, the French microbiologists, were successful in growing them into pure culture medium containing serum and conformed, and they known mycoplasmas. These organisms are unique among the prokaryotes that the lack of cell wall hence lack fixed in their shape and size. So mollicutes are cell wall deficiency gram-positive bacteria. Mollicutes or mycoplasmas are popularly known as cell wall deficiency gram-positive bacteria. Mollicutes have unusually low guanine and cytosine values, usually 23 to 36%. We know generally the DNA is a double helix structure. The DNA is a double helix in structure. Double helix in structure. In this, each helix 20 nitrogen bases or 10 pairs are present. The popular nitrogen bases are, we know, adenine bind with thymine, guanine bind with cytosine. So in this mollicutes are mycoplasmas. These mollicutes are mycoplasmas. They have low guanine and cytosine content. So low GC percentage is very low and 80 percentage is very high. And these mollicutes are separated several types based on 16S ribosomal RNA. Very, very important. The mollicutes or mycoplasmas are separated into different types based on 16S ribosomal RNA. So 16S ribosomal RNA sequence reveals that the mollicutes are coherent and polygenic group closely related to Clostridia and Bacillus. These all mollicutes, the common ancestors. So different type of mollicutes are mycoplasmas are subcategorized. All the mycoplasmas or mollicutes are originated from the common ancestors. The common ancestors, Clostridia or Bacillus. The common ancestors, these mycoplasmas are separated into different types based on 16S ribosomal or RNA. And very, very most important, the mollicutes are gram-positive, lack of the cell wall. So they are resistant to antibiotics. Which type of antibiotics? The antibiotics which destroy the cell wall, they are highly resistant. Why? Because the common definition of the mycoplasma, gram-positive, lacking of cell wall. So there is no cell wall. So these antibiotics, the first one is penicillins, carbaphenins, and cephalosporins or cefamycins, clavams, monobactams, vasomycin, bacitracin. These all are antibiotics to destroy the cell wall and kill the bacteria. But however, in mycoplasmas or mollicutes do not contain cell wall. So these drugs are not work on the mollicutes. So they resist the antibiotics. Which antibiotics? Cell wall destroying antibiotics. Generally, the cell wall destroying antibiotics are belongs to beta lactam antibiotics. Cell wall breaking antibiotics are generally known as beta lactam antibiotics. So molecules are mycoplasma resist to antibiotics, especially which type of antibiotics? Beta lactam antibiotics. Beta lactam antibiotics are to destroy the cell wall of the bacteria. So these in the mycoplasma there is no cell wall, so they did not work. So which are destroy the cell wall. What are there? Penicillins, carbamazepines, cephalosporins, clavams, monobactams, vasomycin, bacitracin. These antibiotics are not worked on the mollicutes or mycoplasmas because they are lacking of cell wall and gram-positive bacteria. And how can we recognize the mollicutes or mycoplasmas? One of the stain, Denise stain, are used for the staining of mycoplasmal microcolonies. And one of the most important, these mollicutes to show the fried egg appearance are nipple-shaped colonies. Fried egg appearance are nipple-shaped colony appearance. So on the culture medium, there are two regions are present. One is the dark region in the center part, and peripheral region is the pale in color and appear as a fried egg. So in the egg, the central yellow part is dark color and peripheral is white color. So the fried egg appearance are nipple-shaped colonies seen in mollicutes or mycoplasma. And another most important, mollicutes growth requirement is depending on sterols. The mollicutes or mycoplasmas are for their growth, sterols are required except Ecoleptomonasteles. Ecoleptomonasteles did not require sterols, but remaining all mollicutes or mycoplasmas for their growth, sterols are required. But one of the chemical, digitonin to destroy this sterol. So all the mollicutes growth is structured when the nutrient medium added with digitonin, but except ecocolnaptinels. So the classification of mycoplasmas. So the mycoplasmas are based on nutritional requirement. Mycoplasmas are divided into genres. So these mollicutes are mycoplasmas are divided into mycoplasmatales, entoplasmatales, Achilioplasmatales, Enereoplasma tails. So in mycoplasma tails, they are aerobic, strongly sterols are required for their growth, and they mainly attack on the vertebrates or posts. But entomopterales, they are aerobic, sterols are requiring, they attack the invertebrates and plant posts. And echoloptomales, they are purely aerobic, but sterols are not requirement. One of the most important, which mollicutes or mycoplasmas are did not require sterols for their growth, echoloptomales. And Eniroptomales, they are completely anaerobic. So in the mollicutes, only one type is anaerobic, that is Eniroplasma tails. So in this mycoplasma tails, one of the best example is mycoplasma. So several years, the scientists are very controversial mycoplasmas and mycoplasma. Mycoplasmas and mycoplasma. That's why the mycoplasmas are converted into a changed another synonym name mollicutes. So mycoplasma is the part of mollicutes or mycoplasmas. And in our syllabus, there are two. One is spiroplasma, and another one is phytoplasma is present. So in our syllabus, which are present, one is spiroplasma, spiroplasma, and phytoplasma is in our syllabus. Spiroplasma belongs to Spiroplasmataceae and Enteromorphales order, Entomoplasmataceae and family Spiroplasmataceae and genus Spiroplasma. And Ecoliplasmataceae are again two families, and Ecoloplasmataceae and Insecticides. In that phytoplasma is belongs to Clinoplasma tails. So mollicutes, that there are two type studies are within Spiroplasma and phytoplasma. Spiroplasma belongs to Entomoplasmataceae. Spiroplasma belongs to Entomoplasmataceae, and phytoplasma belongs to Ecoliplasma tails. Most important, phytoplasma did not require sterols for their growth, because phytoplasma belongs to Ecoliplasmataceae. Ecoliplasmataceae did not require sterols for their growth. But however, in spiroplasma, sterols are required for their growth. So first, we learn phytoplasma. Several years, these organisms did not give their name phytoplasma. Simply they known as MLOs, mycoplasma-like organisms. They are very microorganisms. They are filtrated by bacteria, filter paper. So they are resembled to mycoplasma. So a lot of years, the scientists give the name MLOs, mycoplasma-like organisms. So phytoplasma is popularly known as MLOs. M means mycoplasma, L means like, O means organisms. So these phytoplasmas first discovered by Japanese workers Dio et al. And these phytoplasma, or whatever it may be, spiroplasma, both are almost similar characters. Phytoplasma and spiroplasma both are attacking the phloem part in the higher plants. So phytoplasma and spiroplasma both are affected the phloem part of the plant, especially sieve cells and sieve tubes. Why? Because the growing of the phytoplasma or spiroplasma require the sucrose. The sucrose part is present in the sieve tubes are the higher plant parts. So generally the phytoplasma or spiroplasma is located in the phloem parts. So these phloem parts affected with certain diseases in the plant body. So these phytoplasmas and spiroplasmas are carried by insect vectors. Most important, the phytoplasma and spiroplasmas are carried by insector vectors. So phytoplasma cause the plant yellow diseases. Phytoplasma causes the plant yellow diseases. And phytoplasma basically survive in phloem tissue plants. Phytoplasma as well as spiroplasma also basically survive in phloem tissue, especially in sieve tubes. Why? Because their growth is required for the sucrose. So non-cultured sieve tubes are restricted mollicutes, phytoplasmas, they cannot be cultured. So phytoplasmas are non-cultured sieve tubes, restricted mollicutes. The most important, phytoplasmas are cannot be cultured into artificial or synthetic medium. So non-cultured sieve tubes, restricted mollicutes are phytoplasmas or MLOs, mycoplasma-like organisms. So recently, different type of plasmas are given temporarily. So the phytoplasma taxonomy group proposed to accommodate phytoplasmas with the novel genus Candidatus phytoplasmas. So phytoplasmas nowadays we called Candidatus phytoplasma. Simply CA phytoplasmas. That means CA means Candidatus phytoplasmas. These Candidatus phytoplasmas are transmitted by insect vectors. Most important, they carry the one plant to another plant by insect vectors, especially suckers, mainly phloem feeding leafhoppers. Phloem feeding leafhoppers of family Cicadillae. Cicadillae and plant hoppers of the family Cixilidae and Psyllids and Psylloida. So most of all phytoplasma and spiroplasmas are transmitted by insect vectors popularly known as leaf hoppers and plant hoppers. Leaf hoppers and plant hoppers are transmitted insect vectors from one plant to another plant. So factors associated with phytoplasma infection, low level of indolestic acid oxidase. So in the plant body, IAA is decreased. Indolestic acid, this is the natural first synthetic discovered auxin or phytohormone. If the IAA oxidase is decreased, then the plant easily susceptible to phytoplasmas. A lot of diseases caused by phytoplasma in flowering plants, coconut lethal yellowing, caused by phytoplasmas. Nowadays, phytoplasmas are called Candidatus phytoplasma. And peach X disease and little leaf of brinjal. Most important, little leaf of brinjal is caused by phytoplasma or MLO, mycoplasma-like organism. Grassy shoot of sugarcane and phyllodes of sesame. Phyllodes means phyllodes of sesame. Phyllodes means generally the leaf and some of the calyx parts are green color. Remaining floral parts are non-green color. But however, the floral parts are converted into green color. Calyx, androecium, and gynoecium. These are colorful parts, and the sexual organs are converted into green leafy-like structures. Now they are called phyllodes. And sandal spike, one of the most important sandal spike is also caused the diseases by phytoplasma. Now it is called Candidatus phytoplasma. Witches brooms, another most important disease. All witches brooms caused by phytoplasma. One of the best example, first disease discovered caused by phytoplasma, aster yellow witches brooms. Aster yellow witches brooms. Aster yellow witches brooms, phytoplasma. Now the name is Candidatus Phytoplasma asteris. It is the first discovered phytoplasma diseases. Candidatus Phytoplasma asteris, aster yellow witches brooms. So most important, little leaf of brinjal and phyllodia of sesame, sandal spike, witches brooms caused by phytoplasma. And it is most important, they did not grow in culture media, non-culture, and it is attacked the phloem parts, especially sieve tubes. And another one type of mollicutes, spiroplasma. The spiroplasmas belongs to the class Mollicutes, order Entomoplasmataceae, family Spiroplasmataceae, and genus Spiroplasma. This spiroplasma is appeared in spiral shaped structure without cell wall and gram-positive bacteria, and they show the helical-like structure. Most important, which molecules are to show the helical-like structure? Only one, that is spiroplasma. Spiroplasmas to show the helical structure and show the motility So spiroplasmas and phytoplasmas belongs to molecules in that the group unique bacterial organisms characterized by small genome sizes, low GC content 23-40%, and lack of the cell. So spiroplasmas are correlated with phytoplasmas in the tissue, low GC content and lack of cell wall, and both are gram-positive bacteria. And their common ancestor is Clostridium or Bacillus. So the plant pathogenic molecules are mainly restricted to phloem tissues of the plant. So spiroplasma are also to attack the phloem parts in the host plant. So the plant pathogenic molecules all are transmitted by insect vectors, mainly phloem feeding leaf hoppers and plant hoppers. So almost all same. The phytoplasmas and spiroplasmas are transmitted by insect vectors, sucking in vectors, leaf hoppers, family Cicadellidae, and plant hoppers of the Cixidae and Psyllids and Psyllid. And most important, the spiroplasma, not all spiroplasma, some of the spiroplasmas are to release the one protein. That protein is spirulin. Most important, spirulin. This spirulin is the abundant protein and major surface of antigen. So the spiroplasma secreting the some antigen. That antigen is spirulin. And spiroplasms have unique style movement, and spiroplasms do not have flagella. Most important, the spiroplasms do not have flagella, but have internal cytoskeleton and are motile. The spiroplasmas are contain intercytoskeleton and are motile. Spiroplasma intercytoskeleton is flat and membrane bound ribbon composed parallel fibrils. The fibril ribbon binds to the inner side of the spiroplasma cytoplasmic membrane, follow the shortest helical line and extends the entire length of the helix. So this is the spiroplasma, and the different shapes are present, which is in spiral manner, and inner side of the cell membrane, inner side of the cell membrane it composed of a parallel fibrils. These parallel fibrils to show the movement. So then they have moved one place to another place and also change their shape. So spiroplasmas do not have flagella, but they are motile in the presence of internal cytoskeletons. Most important, internal cytoskeletons. Internal cytoskeletons are clot and membrane bound ribbon composed of parallel fibrils. So among these spiroplasmas, only 3 spiroplasmas are plant pathogens. Only 3 spiroplasmas are plant pathogens. One is Spiroplasma citrate. Spiroplasma citrate. They cause the citrus stubborn disease, CSD. Citrus stubborn disease caused by Spiroplasma citrate. Spiroplasma kunkeni. Spiroplasma kunkeli. Corn stunt spiroplasma. CSS. CSS. Corn stunt spiroplasma. So they cause the disease corn stunt spiroplasma Gia maize. Gia maize, sweet corn. So corn stunt spiroplasma CSS caused by the spiroplasma kunkele. They attack the Gia maize. Spiroplasma citrine to attack on the different type of citruses, sweet citrus and loose orange and different type of all citrus species cause the spiroplasma citrine. The disease name is citrus stubborn disease. And spiroplasma phoenicium, periwinkle yellow disease, PYD, periwinkle yellow disease caused by spiroplasma phoenicium. These 3 diseases are caused by spiroplasma on higher plants. These are only 3 spiroplasmas or plant pathogens. That one is Spiroplasma citrine, Spiroplasma kunkeli, and Spiroplasma phoeniosum. CSD, citrus stubborn disease caused by S. citrine, on the citrus species belongs to Rutaceae family, and corn stunt spiroplasma CSS caused by S. kunkeli, and periwinkle yellow disease PYD caused by S. phoenicium. And the plant pathogenic spiroplasmas are restricted to sieve tubes of their plant host and are transmitted from plant to plant by phloem feeding leaf hoppers like spiroplasms.